So here's a little cartoon of the gene made up of all these bits that get stuck together, and then that makes an RNA, and then that codes for this transcription factor protein. So the red bit is this homeodomain that Kevin talked about earlier. This is the bit that actually binds to DNA. The yellow limb domains, once this is, because it's not a linear thing, it folds around on itself. The limb domains interact with other transcription factors so that a whole complex forms to allow for gene expression. Here's a cartoon, and as of last year, those are all the mutations that had been described in Neil-Patella syndrome. All of the ones down here are spelling mistakes. So a protein is made, but it just doesn't have normal function because one piece was changed. So all these changes here are going to stop it from binding DNA, and any of these changes here probably affect how it interacts with other proteins. All the symbols in red up top will stop the protein being made at all. So with Neil-Patella syndrome, you have half the normal amount of LMX1B, or half the normal amount of LMX1B function. Either you have half the normal amount, period, if you have one of these mutations, or you'll make a normal amount of LMX1B, but only half of it works if you have one of these mutations. Some people are missing the gene completely, and I won't try and explain this whole figure, but you see in these people with stars, you can see how these blobs are much fainter than in the others, and that's because they're missing the whole gene. And it doesn't matter whether you're missing the whole gene, you have one of those red mutations that stop protein being made, or you have one of those purple mutations that allow the protein to be made and it doesn't work. That any of those mutations mean is that you have Neil-Patella syndrome. It doesn't mean that it's mild or severe, or it's with kidney disease or not, or with glaucoma or not. Whether you get glaucoma or not, or whether you get kidney disease or not in the context of Neil-Patella syndrome is due to something else. It's not the particular mutation of LMX1B that is causing you to have kidney disease. The mutation in LMX1B causes you to have Neil-Patella syndrome. How severe your Neil-Patella syndrome is relative to somebody else in the room or a relative of yours is to do with other factors involving other genes, and the identities of those, some of them we could guess at, and others we have no idea. So this is just all the different types of mutations that cause Neil-Patella syndrome and resulting in the same range of symptoms. The mechanism that in molecular biology that we use to describe this is called haploinsufficiency. So haplo is half, so having half of the normal amount of function is not enough for normal development. So I'll just summarize that. LMX1B is a transcription factor. It's expressed in the tissues affected by NPS. It is caused by loss of function mutations in LMX1B. So I'll get into that in a minute, but what I want to say, or what I think we'll maybe touch on this later in the question and answer session with Dr. Tierney. But you're talking about when those of you who have fibromyalgia, that is, and chronic fatigue syndrome, that is regarded mostly now as being a serotonin deficiency. And so when all of your irritable bowel syndrome and GI issues, when I showed you those embryos, there was no LMX1B expression in the abdominal area at all. LMX1B is not normally performing a function in that area. So your symptoms between basically here and here are due to consequences of it not being expressed elsewhere, and that the signals are not making it to the other parts of the body. So I've been asked to discuss some common questions and misconceptions, and hopefully these next few slides will clarify for you. So remember what I said this morning, NPS is inherited as a dominant trait, or dominant phenotype. So affected males and females in multiple generations This is what we call vertical transmission, because when you pull the pedigree from the wall, it goes vertically. Each affected individual has a 50-50 chance of passing it on for each child. It does not skip generations. What you said about if NPS was in both parents, then because it's a 50-50 chance whether dad here passes on normal or NPS, or mum passes on normal or NPS, a quarter of them would get what we'll call double NPS, a half would have regular NPS, and a quarter would be unaffected. So here's a situation. We've got someone with NPS, and they've got a couple of kids, and they thought they were a new mutation because they didn't have, neither of their parents was affected, but their sister has unusual nails. What might be going on? Well, could be dad might have been affected and it was mild and they hadn't noticed. Or there's a condition, if you have the mutation in some of your cells and not others, then depending on whether it gets into your germline or not, then it can be passed on or not. So that's a possibility. Or it's just coincidence. There are many features of NPS, as we've discussed, that can occur by themselves or are part of other conditions. So fifth finger clinodactyly, club foot, ADHD. All of these things are way more common as isolated findings in any member of the population than Neil Patella syndrome. Showed you club foot, the incidence is about 1 in 1,000, so at least 30 times more common than Neil Patella syndrome. Fifth finger clinodactyly, probably about the same. ADHD, something, let's say 5%. So a lot of times more than Neil Patella syndrome. Yes? Oh, sorry, a mosaic is where you're composed of two different cell types. So if a mutation occurs early in development, in the first few cell divisions after conception, then some cells have the mutation and some don't. And depending on which cells, or which part of the body those cells end up in, somebody may or may not have symptoms of the disease. Okay? So spontaneous mutations or sporadic cases, this is a new change in DNA, as I said this morning, where the change in DNA occurs during the differentiation of cells to become either sperm or eggs. And so the person in which that mutation occurs, it occurs in their germline. They have no clinical consequences for that mutation, but it could be passed on in a sperm or an egg to the next generation. The single base substitutions, so that's all of the which are by far the most common cause of mutation, those occur more often during sperm development than egg development. Deletions of the whole gene or rearrangements of the gene occur in either, but many of these small mutations are more likely to occur in sperm development. And some sequences are more susceptible to change than others. These are called recurrent mutations. The same mutation occurs over and over again. Those of you who have had your mutations identified, talking to each other, you both may have the same mutation and you never knew you had a common relative. You probably don't. It's the same mutation occurred by chance in unrelated individuals, just because certain sequences are more susceptible to change than others. What are the advantages of genetic testing for Neil Patella syndrome? First, it can confirm the diagnosis. In 90-something% of cases, Neil Patella syndrome is a straightforward clinical diagnosis. The pictures I showed you of the nail changes this morning are really very distinctive, and anyone who's seen them once or twice should be able to diagnose Neil Patella syndrome clinically. If they're not so sure, certainly it's possible to do a genetic test, and by identifying an LMX1B mutation, that would confirm that it was Neil Patella syndrome. It is more likely that you'll get an accurate clinical diagnosis than find a mutation, Mutations will only be identified in 90 to 95% of cases. I would expect a competent clinician to make the diagnosis more often. If they've seen a case before, they should be able to recognize it a second time. A positive test would allow for confirming the diagnosis, so you would initiate early testing for kidney and eye disease, eye complications, and that would certainly be worthwhile. If it's negative, then it sets your mind at rest, so that's always good. It does require, you have to identify the mutation, or there has to be a strong family history. You have to have like one of those, more than that. No, this one. Like a number of affected people, so then you can determine, so if they have another kid, is it affected or not? You can do it by tracking genetic information through the family, even if you don't know the nature of the mutation. And we talked about prenatal diagnosis, and this was one that we did before the gene was actually identified, but we knew that this characterized this particular part of chromosome 9, and we could see that this part of chromosome 9 had been passed to his affected son, and then they had a series of prenatal cases.
Well, at least one person in a generation, at least, that will get it. It'll present. Is that...
No. If you have NPS, you have the first child, it's a 50-50 chance whether they get NPS or not.
Right.
Second child, 50-50 chance. Third child, 50-50 chance. So each is an independent event.
Right.
And so if you don't pass it on, they won't pass it on.
Oh,
okay. Okay? So, and then in the end up, they had a couple of twins that didn't have it, so they were happy then. And that is where I will stop for this. Is there any other questions on this? Yeah.
You mentioned you're talking about disruption of serotonin.
Mm-hmm.
Is there a correlation between that and NPS and people having sleep problems, or is that?
Normal serotonin and normal dopamine levels are what stop you having depression. Those are what doctors try to adjust for people who are depressed. A major part of depression is disrupted sleep. So if you've got disrupted, if someone's been worked up for depression, and the others will talk more about this later because they're experts and I'm not, having a disrupted sleep cycle is part of being diagnosed with depression. But they'll talk more about that later on, I think. Okay, no problem.